For this opportunity, I would like to share some experience from the national program called Genomics Thailand. So I would like to begin with an introduction of our national program called Genomics Thailand. So you might recognize these names. It's pretty similar to Genomics England in the UK. So we use this as our inspiration to establish also our national program. But the aim is to implement this genome technology and genomic medicine in the healthcare system. With the vision, we aim for two-pronged approach. First is to try to accelerate or facilitate the application of genome technology into the healthcare system. In the meanwhile, we use that opportunity to collect the data to build the population-level genome database. With the aim on the first phase of Genomics Thailand, about 50,000 people. And with the patients or any individuals who have a various kind of indication of testing, for example, cancer, rare disease, pharmacogenomic testing, infectious disease, or even chronic disease, or even established healthy cohorts. We use that opportunity to do not only for a targeted gene sequencing by panels or by single gene testing, but we also collected to do a whole genome sequencing to build those databases, and hopefully with this kind would be a great resource for the future use, either both for diagnostic and also research. And obviously, any positive testing from those would be delivered back to the healthcare professionals. And I'm overseeing just one part of Genomics Thailand, which is the cancer program. With this one, we also use this opportunity to also analyze the data and use that with the other metrics on the treatment to study on cost-utility studies and try to make sure that those tests would be ready to integrate into the national healthcare program, because our healthcare scheme are mainly covered by what we call universal health coverage. We use this opportunity to try to push forward and make these tests reimbursable as much as possible. And for this result, since the beginning of last year, the first test for BRCA testing would be eligible for all Thai patients who have a clinical indication for free. And with that opportunities, we collect up until now, in the third year of the program, we are about halfway through. About 25,000 individuals have been collected. More than 7,000 individuals are cancer. About more than 5,000 patients has been analyzed, and surprisingly, although about 1 in 6 individuals indeed carry a significant pathogenic or likely pathogenic variants. It's pretty high compared to the Western populations. When we look at the different genes that we analyze, you see that obviously BRCA1 and 2 account about 40 to 45% of the cohorts, obviously, because the majority of the patients are either breast or ovarian cancer. But what we see is about 6% of the patients who indeed carry pathogenic or pathogenic are CNVs. And if you are familiar with the clinical interpretation of those CNVs, you think that the framework of clinical interpretation is very challenging, because there's no established way to call it pathogenic or pathogenic very easily. For example, if you have those kind of genes that known to be a haploinsufficiency, to be a cause or pathogenicity of those disease, when you have a deleted segment, it will be easy to interpret as pathogenic. But the problem is with short-read sequencing, you don't know what's the extent of the deleted segments. But for the duplicated segments, it's become very difficult to interpret. Either it's pathogenic or it's just a insignificant, even benign duplicated segment, because we don't know what the location of those duplicate segments is. If it's in the genes, it's likely to be disrupted, the function likely to be disrupted. You can call it pathogenic variants. But if those duplicated segments, it's located outside the gene or far away from the real genes, so it's probably no significance at all. So it's become challenging when you try to deliver those interpretations to the clinicians, because positive or negative findings on those test results have meaningful impact. Either the doctor decide to pursue with the rigorous surveillance or to do a preventive measure, or you just live with nothing. You don't need to do anything else. So for this, I would like to give you just 2 examples of the case that we have. The first one is the young patients who come to see us with colorectal cancer, and with the colonoscopy, we could identify the several polyps throughout the colon. So with the clinical diagnosis would be very easy that the patients have colorectal cancer that caused by the called familial adenomatous polyposis. With the targeted short-read sequencing and the CNV analysis, we can easily demonstrate the whole gene sequence, whole gene deletion in the APC gene, which is a causative gene of this condition. But the problem is not the diagnosis that the patient has, but how big of this deleted segment. We don't know. We can confirm it with the digital MRPA to see the deleted segment entire genes, but we don't know the extent at all. With this beauty of adaptive sampling, reject that read and map back to the whole genome, we can see the coverage drop that is quite huge, about 5 megabase, that span even more than the APC gene into other genes as well. And some genes are clinically meaningful. For example, you see the gene SLC 25A46. This is one of the genes that cause peripheral neuropathy. So the patients does not only have the colorectal cancer, which is inherited, but also have another genetic disorder that we have to look for once we successfully treat her, for example. And the second case is the patients that come in with pretty common problem, breast cancer. And she has a several family member, which is a first-degree relative. It could be at risk if we can identify this pathogenic variants. But the problem is also, when you look at the CNV analysis, you see duplication. And duplication is always problematic. You don't know if it's really pathogenic or not, and you have 2 duplicated segments. One is APC, which is totally unrelated. And the second is CHEK2, which is might be a causative gene of her condition, which is a breast cancer gene also. But how do you know if this duplicated segment or partial duplication of this CHEK2 is pathogenic? If it's not, then we don't need to do anything else. We can ensure them that this is likely to be a common cancer. It just happens once in a while, and other family member could be at risk, just similar to general population, or the entire family are at risk who indeed carry this duplicated segment. So again, digital MRPA confirmed the presence of duplicated segments. With the long-read sequencing, we are able to identify that indeed duplicated segments reside in the genes. So with this code, it's likely that this partial segment duplication would be disrupted to the gene function and is likely to be a pathogenic in this case. Meanwhile, the APC partial duplication actually is caused by the fusion segment that involve 2 genes between APC and the other gene, and this segment sit outside the genes, the APC. So the entire APC is still intact. There's no disrupted function of those. So we can conclude that this one, the CHEK2 partial duplications, is likely to be a pathogenic in this family. So With this, just give you two examples of the real case that we encounter, and we can see that with the regular short read sequencing from the target sequencing in the cancer program in our Genomics Thailand could identify some potential limitation of those, and how long read sequencing like nanopore could help us resolve those mystery. And with this work, I'd like to thank a lot of people to that tip from Sirat Longlit Lab. She spearheaded the long read sequencing in our program, and other scientists that working in the lab, and the huge program that we would like to expand more. And if you have any questions, feel free to discuss further. Thank you very much.